go back to the middle so I can point it out. So I guess your question is, if we're looking up here, if you look at it long enough, and we just look at this structure, I can talk you into 2 points of view. And I can say, well, imagine this is here a valley. And what we're looking at is that the shadowing came from the left, and this is a valley. And so that's our point of view. But we can also say, no, this actually is a hill, it's a ridge, and the shadowing came from the other side. And if you look at it long enough, you can convince yourself of both points of view. So then you have to decide which point of view is correct. And then you look at the very small particles which have to stick out. So where they have their shadow, that reveals where the shadowing source is. And based on that, you then ultimately decide that right here, this has to be a valley. And then that means that right here, I'm now on a hill.
That makes sense.
Yeah. Okay. A fibroblast is a connective tissue cell, and this is scanning the EM image. Once again, we look at something that looks like an object in three-dimensional space. It's not like we're looking down on a landscape. So this is just what I wanted to show you as the contrast between these two. It looks really like we're looking at something that occupies the entire surface, and here we have a three-dimensional object in a space. Okay, another negative staining image for a virus. So we now know how that is made. There's another virus. This is a virus that can infect us. It's another negative stain, so you can see the spikes sticking out. So based on a structure like this, you can think that the virus will look something like this drawn structure here. Again, and finally, I have two thin sections to look at. So this is clearly a mitochondrion, and you should recognize this type of structure as a mitochondrion, because you should know the main structures of the main organelles of the cell. We also see a lot of ER in here. And this is a very crisp and nice image. And it's an animal cell from which this section has been taken. And animal cells are easy to fix. In contrast, plant cells, which tend to be bigger, and they have a nasty cell wall, it's much harder to fix them, whether you try freezing or chemical fixation. The fixative comes in slower. You get the temperature to go down slowly if you do that approach, or if you do chemical fixation, you fix it more slowly. So this image of part of a plant cell with a chloroplast is a little bit more blurry than the image here of our mitochondrion, which is very crisp when you look at the membranes. Now these membranes here that we see in the mitochondrion, or that we see here in the chloroplast, are the same thickness. The only reason that we have a little bit less definitional structure here in the chloroplast is it's more difficult to fix. You get a little bit more blurry structures. And I just want to tell you that because we need to understand that the quality of the image that we see, especially in the EM, depends on how we prepare the sample, and sometimes it's just hard. Okay, now that's enough about these. And let's go and see if this one, okay, this one wants to work for me. So, if I do another one, woohoo, that works. Okay. So we've looked a lot at transport systems. Remember, we have different types of transporters. And what I think is the type of level of understanding that you should have is a level of understanding that allows you to predict what would be going on. So let's just say that we have, a vacuole. This is a vacuole. And we're having an organism that for some reason wants to put inorganic phosphate, so Pi, into the vacuole. Or it could be a lysosome. And inorganic phosphate has a charge of, we can expect a charge of -3. So now you can ask yourself the question, what would I expect for a mechanism by which your cells take up this phosphate and put it in this lysosome? The lysosome is the vacuole of the mass. Okay, so what do we remember about the vacuoles? Is the pH low, high? We have about a pH of, well, shall we call it 4? That's pretty low. It might not be that low. But we have an excess, therefore, of hydrogen ions, right? And we have a neutral pH in the cytosol, so that's outside, so we have a low concentration of protons outside. So how did the cell achieve this difference in proton concentration? What type of transporter or mechanism was that? We're moving protons into a compartment where there's already a lot, then we have to use a pump, right? So the protons came in by means of a pump. Something that probably used ATP. A pump. I'm left-handed, but I have to use the mouse right-handed because that's where it's connected. Challenge. Hey, you have to make the exam on Friday. It might be difficult for you. Okay, will we have a membrane potential here? Almost always is the answer yes. Okay? Is the membrane potential such that it is negative or positive inside the vacuole? It's positive. That's great, you got that. Now that's really important. It's positive in and it's negative out. Cool. Okay, so now is the movement of phosphate favored by the membrane potential? Yes, very favored, because there's free charges on the phosphate that are all pulled in. If phosphate accumulates in the vacuole, that means if the concentration in the end is higher in the vacuole than in the cytosol, would that movement of phosphate be favored by its own concentration? Probably not. But if we think about the effect of the membrane potential here, and the fact that phosphate has so much charge, would it be possible to use a uniporter to get the phosphate in? Yes, that is possible. Would it be possible to use a channel to get the phosphate in? Yes, that is possible. Would it be efficient to use a pump to move the phosphate in? No, it's not efficient. It is theoretically possible, but it would be very inefficient. So as a likely mechanism, you would predict that we would have a transporter here, which is either I'm not going to write it, either a channel or a uniporter. Now, if we want to accumulate an awful lot of phosphate in here, then maybe an anti-port system might be required, but that's only if you need an awful lot. Now, this was inorganic phosphate. Let's just say we want to move ammonia in. So ammonia, I'm going to suffer and I'm going to draw ammonia. And it's positively charged. Okay, we want to accumulate ammonia in the vacuole. Is that favored by the membrane potential? No. is that favored by, if we're accumulating it, so we're stuffing the vacuole with ammonia, it's not favored by concentration. So can we go by means of a channel? No way. Can we go by means of a uniporter? Also not likely. So actually, from the point of view of how you drive the transport, a channel or a uniporter, they're driven by the same. It doesn't matter. It's just that the uniporter will be relatively slow compared to the channel. The channel will be maybe less selective, but it will be super fast. So channel and uniporter are excluded. So what is your best guess for the uptake of ammonia if you need to do that? You could use a pump, but that is inefficient. You're using an antiporter with which ion? You use hydrogen ions, right? Protons. So you're going to really predict that we're going to have here an antiporter, and we're going to have transport in exchange for, this is my arrow, in exchange for a proton. Okay. That's what you predict. You can reason yourself through this. Similarly, you can reason yourself through that very nasty, pesky epithelial transport system. Such as, for instance, here we have the small intestine, epithelial cell. And, okay, you have to suffer through my joints, okay? You cannot do any better. Okay, that's an epithelium. A cell, another cell. We have here the tight junction. Isn't it great? You can look this back up on the echo system. It's all recorded. I cannot say bad things about Mr. Putin. The evidence will all be there. Okay, so we need to get glucose into our body. This is the lumen of the gut. So we're saying here that is the lumen of the gut. You've just had your lunch, you've digested all the starch, you've digested enzymes, and that starch is broken down to glucose. You're now taking up that glucose because you have good pasta, and that's now converted to glucose. You want to get the glucose in, and that's an in arrow, and you want to get it out here into where we have the bloodstream. The blood, okay? Enough. So, how do we do this? So we have a low concentration because your lunch is digested slowly. We have a low concentration of glucose here. We will have a high concentration of glucose in this cell. So to get our glucose in, this is not favored because of concentrations of glucose. Now, glucose itself is not touched by the membrane potential. The membrane potential is, by the way, always positive out and negative in. And that means we're also here positive out and negative in. We're here on the apical side and we're there on the basolateral side. Remember that. Separated by the tight junction. We cannot have transport here through the tight junction. So glucose has to come in. Well, it has to have some help. It gets help from our friends, the sodium ion. Sodium comes in. Actually, it needs a lot of help, so 2 sodium will have to come in. This is clearly a 2. 2 sodium ions come in together with glucose. Okay. Well, if we get now we're just going to reason ourselves through what we need. And we remember we need 4 transporters. 2 sodium come in. Well, pretty soon our cell will be stuffed with sodium, so we need a way for sodium to get out. So we have here our P-class pump, and it takes 3 sodium and A out. Wow. And it takes at the same time it takes 2 potassium in. Okay, 2 potassium go in, and we're hydrolyzing an ATP there. So there's ATP, oh, this is terrible, right? An ATP to ADP hydrolysis is required for that process. All right, well that's nice. Now, this cell then exports its sodium, but now it stuffs itself with potassium, so potassium has to get out. We need a channel for potassium. This is a, wow, bad drawing. Potassium flowing out through a potassium channel, okay? We have a channel there. Okay. Channel. That's a channel. Oh, glucose was accumulated because of the action of the sodium here at high concentration in the cell, so glucose can go out there via a uniporter, just down gradient. Export of glucose here is favored by the glucose concentrations. It's not touched by the membrane potential. Sodium-potassium exchange is driven by ATP hydrolysis. Potassium flows out through the channel favored by its own concentration because remember, potassium is a dominant ion in every cell. So it flows down gradient. Potassium outflow is not favored by the membrane potential. But there's so much potassium accumulating inside it gets out. Here, glucose uptake is driven by sodium uptake in symport, co-transport. Sodium uptake is favored by the membrane potential. Sodium uptake is also favored by sodium concentrations. The sodium concentrations are kept where they should be by means of this all-important pump. You can reason yourself through the entire problem. You always can. If you can, you're good. Okay. I'm sorry this board didn't work the way it should have.